We're going to do a running tour of microbial diversity today. So we're going to talk about, continue on the topic of microbial diversity. We probably won't quite finish this topic, so we'll probably cover this a little bit in the next lecture. And then the remaining lectures on that I'll be doing will be on different sort of functional and biological topics about microbes. And the first one that we're going to do is talk primarily about modes of reproduction and how they acquire traits from other organisms, either through stealing genes or through symbioses. So I have these four categories of diversity that we're talking about. We've already talked about the first two, the diversity of form and the diversity of movement or motility. And what we're going to do today is talk about the diversity of function and the phylogenetic diversity. And again, we're sort of taking this big picture approach. Really, in a way, we're covering all of the organisms on the planet, although I'm sort of leaving out plants, animals, and fungi because those will be covered separately in the remainder part of the course. So we're going to focus broadly on diversity of the other lineages. So in terms of function of microorganisms in particular, many of you, I am sure, are familiar with a variety of microbes through the nasty things that they do. So microbes are the causative agents of many diseases. Certainly bacteria cause a lot of diseases in humans, plants, and animals, whether that's tuberculosis or cholera or anthrax or dysentery or all sorts of other things. We will talk a little bit about viruses. So like I said, we're not really including them on the tree of life, but we will talk a little bit about viral diversity, and viruses, of course, cause a lot of nasty effects on organisms. And there are many microbial eukaryotes that also cause a variety of problems. So these are the microbes that most of the world sees, and it's, in many cases, the microbes we know the most about their biology because they've been studied in terms of how they make things sick or how we can kill them. And people have learned quite a bit about their biology because of their negative effects, usually on people or animals that we care about or plants that we care about. Of course, actually most of the microbes on the planet, in terms of the total numbers as well as the number of kinds, they're not pathogens. They don't make things sick. Many of them are what are called beneficial symbionts, and we'll talk more about this in a subsequent lecture, but there are quite a few microbes that have beneficial effects on other organisms. And I have a few examples here. So nitrogen fixation in legumes, which you've done, there's a little bit in the lab about this, and there's a little bit in the book about this. So you can grow legumes in nitrogen-poor soils, and in many cases people grow them as rotation crops and then plow them over and bury them into the soil. And this is because legumes have a symbiosis with bacteria that allows the bacteria pull nitrogen out of the air and convert it into usable nitrogen that the plants are able to use to produce proteins, which are, nitrogen is a key part of proteins as well as other parts of the cell, parts of the organism. Nitrogen fixation is only carried out by microorganisms. So any plant or animal that needs to fix nitrogen in some way has to work with a microbe to do it. Carbon fixation, in a lot of different ways, is primarily carried about by microbes. So pulling carbon dioxide out of the air and converting it into sugars and other usable compounds is largely carried out by microbes, either microbes on their own or microbes that live as symbionts with other organisms. So the chloroplasts, as we will talk about later, are actually derived from cyanobacteria, and that's how plants carry out photosynthesis. And animal nutrition is also greatly aided by a variety of beneficial microbes. So cows and other ruminants that live on grass and plants that have most of their energy locked up in complex carbohydrates like cellulose. Microbes are the things in the guts of these organisms that are able to chop those compounds up and make them available. And there are a diversity of other organisms, animals, that rely upon microbes in some way. Most of the microbes that you see on a plant or an animal, so we'll talk about this in a little more detail, but plants and animals are actually covered in a cloud of microbes inside and outside them. And in fact, many single-celled eukaryotes that we will see examples of are also covered in a cloud of microbes. In most cases, we have no clue what most of these organisms are doing. We're just starting to learn what they're doing. But they're there. They may or may not have beneficial or detrimental effects, but they certainly are part of the lives of many organisms. And we interact with this sort of cloud of microbes that are, let's just call them unknown for now. One of the things that I study and that is of great interest to things like the biotech industry and a variety of other industries is that many microorganisms are able to survive and even thrive in extreme conditions. We'll talk more about this, but these are called extremophiles. There are microbes that grow and thrive at basically boiling acid pools where the pH is 0 and the temperature is near 100 degrees Celsius. There are others like the ones up in Mono Lake where the pH is actually, that should be inverted, that should be a high pH, so very basic environment. There's some that grow above 100 degrees Celsius in the bottom of the ocean where the pressure from the water above is able to keep water from boiling. So it's 105 degrees Celsius, but the water is not boiling there. And there are microbes that live and thrive there. And there are others that like cold and like high salt, and we'll talk more about these. These organisms have been of great interest to industry because inside the cells of these organisms are enzymes that can tolerate those particular extreme conditions. And they're used in things like industrial processes where you might need something to work in those particular conditions. Probably next to pathogens, the other group of microbes that we know the most about are those that we use actively in food production, either sort of accidentally or on purpose. So fermentation processes that many of us like are carried out by a variety of kinds of microbes. Sometimes you may like what the microbes do, like making the moldy parts of some of these cheeses. Sometimes you may not like what the microbes are doing. But in general, we know a lot about these microbes that are involved in food production, so you'll see lots of examples of those. And in general, actually, if you look at the planet and you look at the biological processes on the planet, things like global nutrient cycles, carbon, nitrogen, sulfur, phosphorus, etc., most of these processes are either run entirely by microorganisms or dominated by microorganisms in some way. And when you look at the diversity of processes in these organisms, one way to represent this is through what are called metabolic pathway maps. So this is basically a diagram where the nodes here represent a compound like glucose or lactose or glycine or any other compound, and the lines between these compounds represent an enzyme that's able to convert the compound into something else. And these are what are called metabolic pathways in organisms that start with glucose and end up making something else. If you look at all of the metabolic pathways that have ever been found in any organism on the planet, and you ask what fraction of that is found in, say, animals, it's a tiny fraction. Animals, generally, you could say, I might get hit by some people, but you could say they're metabolically incompetent, and plants pretty much are also. Biochemical processes and the diversity of biochemical processes on the planet are dominated by microorganisms. And if you want to find a new process, this is why a lot of the people who do industrial applications are always looking in microbes to find a particular process, because that's where the diversity of metabolic processes is. And as we will see when we talk about symbioses, is the mic working? Oh, okay. moment of panic there. So when we talk about symbioses, one of the reasons that plants and animals engage in a lot of symbiotic interactions with microorganisms is that that's where the metabolic processes are, like carbon fixation and nitrogen fixation and a variety of other processes. And the plant and animal, in theory, could have evolved that process itself, but why bother if there's a microbe out there that carries out that process that you can engage with in some way and get the microbe to carry out that process for you? That explains a lot of the evolution of plants and animals in terms of interaction with microbes, is finding different metabolic processes that they need to carry out. So we're going to go into some detail in some of these metabolic processes, but in particular, there's one aspect of this that is covered in the book a lot and that we will refer back to throughout the course, in fact, which relates to something called trophy. And what is meant by trophy is really derived from this Greek word to nourish. And what we're talking about is the nutrition of different organisms, where they get their energy and really their food from. And you've seen, I'm sure, these terms all the time in various biology textbooks. So autotrophy, chemotrophy, phototrophy, heterotrophy. They're used to describe the processes for particular organisms. And we can categorize this in a way that makes a little bit more sense. So if we look at the different types of nutrition in different organisms, the main way we categorize this is by three components to the trophy. One of them is the source of energy. So what do organisms use as their source of energy for their daily life? What is their source of electrons? So to carry out many of the processes inside a cell, in particular those related to things like building or breaking down carbon bonds or making a variety of key products, in particular related to their energy metabolism, organisms need to get electrons from some compound or some source that's out there and excite those electrons in some way and then use those electrons to carry out certain processes. So this is a really important component of the nutrition of organisms. And then the third major component is the source of carbon. So where do organisms get the carbon that they use to build carbon backbones to make things like carbohydrates, amino acids, proteins, DNA, RNA, fats, etc.? And when you do this, we're going to go through the main types for each of these components. And really there are two main types that people have identified for each of these major components that describe the nutrition of different organisms. So in terms of energy source, one thing that organisms can use is light. So if an organism uses light as its energy source, that's called phototrophy. The main alternative, is to use chemicals, and that's called chemotrophy. So you can break apart high-energy chemicals and use the energy from them, or you can take the energy from light and use it in some way. In terms of electron source, the one possible source of electrons are inorganic compounds. And if an organism uses inorganic compounds for their electrons, that's called lithotrophy. And if they use organic compounds, that's called organotrophy. And in terms of the carbon source, if an organism uses what are called C1 compounds, those are compounds that have a single carbon in them, like carbon dioxide, CO2, or carbon monoxide, CO, or methane, CH3, and so on. If an organism uses those C1 compounds to build their macromolecules, that's called autotrophy. They are, in essence, fixing carbon. That's the other term for autotrophy. And if an organism uses organic compounds, that's called heterotrophy. So you have these three components, and each of them have two categories. And from that, we can basically describe in a simple way the nutrition of different organisms on the planet. So E. coli, for example, which is this model organism used in laboratories to study the biology of anything, or biology of bacteria in particular, and also a component of our gut community. E. coli gets its energy from chemicals, so you need to feed it things like sugar or other compounds that it takes the energy from, breaks up those compounds, releasing energy, and then it uses that in some way. The electrons that it utilizes are from organic compounds. And the carbon that it utilizes is also from organic compounds. And you stitch these all together, and you get E. coli as a chemo-organo-heterotroph. That's how we describe the nutrition of E. coli. In many cases, people ignore the electron source part of this map, and they just say E. coli is a chemo-heterotroph. Okay, so we can do this now for any organism on the planet, and usually, for many of them, they fit cleanly into one of each of these two boxes for each of these components. There are some organisms that have diverse metabolisms that are able to do many things, and you wouldn't just describe it as a chemo-organo-heterotroph, but many of them are pretty clean. So for example, Cyanobacteria, and in fact, really all plants, because plants carry out their energy metabolism via chloroplasts, which in fact are derived from Cyanobacteria. And plants and photosynthetic algae function in generally the same way as Cyanobacteria. They get their energy from light. They get their electrons from inorganic compounds like water. So they're pulling electrons off of water. And they get their carbon from C1 compounds like carbon dioxide. So Cyanobacteria are photolithoautotrophs. And again, many times people throw away the electron source part of this and just call them photoautotrophs. So when someone says something is photosynthetic, generally what they mean is that it is a photoautotroph. They're generally used as synonyms. So humans, let's try and figure this out. Do humans get their energy from light or chemicals? Right? Do you know where we get our electrons? Yeah, so the food that we eat is where we're stripping off electrons, and you can't live off of just like sulfur. So it's organic compounds. And the carbon source comes from the same thing. So humans are like E. coli, are chemo-organo-heterotrophs or chemoheterotrophs. And you will see throughout the book, when it's describing a group of organisms, for example, or an individual species sometimes, it will throw out one of these terms. This group is mostly photoautoheterotrophs or whatever. Something to that effect. They wouldn't be photoautoheterotrophs, but photolithoautotrophs. Hopefully the book doesn't do that, but I might in lecture occasionally. So what to me is sort of amazing about the diversity of metabolism in bacteria and archaea is, if you look at any of these subcategories, so the chemo-organo-heterotrophs in bacteria, or the photolithoautotrophs, or the chemolithoautotrophs in bacteria and archaea. Within any of those categories, there are frequently dozens of different ways that different organisms carry out those processes. So photolithoautotrophs, there's not just one way to do that. There's actually quite a diversity of ways that organisms acquire the light, the energy from light, and there are quite a different diversity of sources for the electrons as well as for the carbon compounds. There are organisms that live off of carbon monoxide and produce hydrogen gas as a byproduct of their metabolism. There are organisms that live off methane. There are organisms that live off CO2. There are organisms that use all sorts of diverse energy sources. So when you hear something is a photoautotroph, that doesn't mean that that's sort of the mechanisms between two organisms that are both photoautotrophs are not necessarily homologous. This is a general description of sort of the phenotype of organisms and not, comparing them. You have to look at the details of how they're carrying out those processes. So that's sort of the backbone of functional diversity. We will see when we talk about extremophiles and other organisms, we'll see more discussion of this, but I wanted to give you sort of the backbone, and you really have to understand these different forms of trophy. Are there any questions about this before moving on to our 30-minute tour of phylogenetic diversity? So what we're going to do now will be a little painful, perhaps. Your head will hurt a little bit. We are going to walk our way through the phylogenetic diversity of bacteria and archaea, and then get into the beginning of the phylogenetic diversity of eukaryotes. When we do this, you should learn the names of the major groups of organisms, and certainly for the big lineages, the key properties of the groups. We're basically covering what is in chapter 26 and 27, so you need to read that part of the book and try and learn this. We're just going to do a quick tour. In terms of studying, what I would recommend, and I will put out another study guide, a study guide for this, is I would recommend working at the root of the tree first and going out towards the tips. So what is common across all organisms? Those are the universal homologies. And then that's, the real root of the tree. And then there's the bacteria, archaea, and eukarya. What are the features that are either shared by all of those or unique to one of them versus another? What are the major features of bacteria versus eukaryotes? And then continue to work your way out towards the tips. So we will see 6 major groups of bacteria, 2 major groups of archaea, and a lot of major groups of eukaryotes. And just do it that way. I'm not going to expect you to know individual species unless we spend a lot of time on them. So work your way out from the root of the tree to the tips and try and learn what are features of the real major groups. Yeah?
We have a great activity in the CLC for you to do this on one of the tables. We have a huge tree, and in the plastic boxes we have characters, so you can practice putting the characters on the tree.
Yeah, good. All right, so we're going to do this quick tour again. This overlaps really completely with what's in chapter 26 and 27. So for the bacteria, in the book, there are 6 major lineages of bacteria shown on this tree and discussed in more detail in the text in the book. In reality, there are many more lineages of bacteria. This is from 10 years ago. There were 40 or so phyla of bacteria that had been formally described 10 years ago. Now there are about 200. We are covering 6 of them. If, be thankful we're not covering 200. 6 is still, I know, a little bit overwhelming, but the diversity here is just extraordinary. And we know very little about many of these groups. As you will see in a few lectures, most of them are only known from environmental samples. But we do know a lot about some of the groups that we're not going to talk about. And what we're going to do is cover these 6 major lineages like they do in the book. These are probably the best studied phyla of bacteria, largely because there are key taxa in each of these groups that are of interest to humans. So we're just going to walk through each of these groups now. So the first one is called the Spirochetes. The Spirochetes are all gram-negative. They have that general structure of gram-negative bacteria like we outlined before. Many of them, most of them probably, are motile, that is, they can move around. There are bacteria and archaea that don't really control their movement much, but these ones, many of them are motile. Most, if not, I think all of them are chemoheterotrophic. And one of the really interesting things about Spirochetes is that they have a flagellum, like many other bacteria and archaea, but the flagellum is inside of the cell, and it's anchored inside sort of the membrane of the cell. And so when an organism has a flagellum that's on the outside and it spins it around, the organism sort of swims around, like in that video that I showed you. When Spirochetes do spin around their flagellum, the cell screws, like a corkscrew. And this is unpleasant if you are infected with one of these organisms. That's how they sort of move around inside the bodies of hosts. Very unpleasant. They are well studied because they include the causative agent of things like syphilis and Lyme disease. Most of them that are characterized are pathogens, but there are some that are free-living. So again, look in the book. You will see some discussion of spirochetes in the book. We're now going to march our way on to another lineage. The Chlamydia group, as many of you may unfortunately or unfortunately recognize the name, the Chlamydia group is well studied because they include the causative agent of many diseases, including the sexually transmitted disease named after the group, basically. But it also includes this causative agent of trachoma, which causes blindness in a lot of people, and the causative agent of some forms of pneumonia. They are also gram-negative. Some of them are cocci-shaped and some of them are rod-shaped. And one of the really confusing and interesting things about some of these is that many of them can alternate between a cocci shape and a rod shape. And this led to many mistakes when people were studying infections caused by these organisms, where you would look in a microscope and you would see things that were rod-shaped and things that were cocci-shaped. And people thought this person has an infection with multiple organisms, when really it's just a single thing that switches shape. They are only known as parasites that live inside the cells of other organisms. And they are extremely small. In fact, for a long time, chlamydias were characterized and classified as viruses because they're so small. When people were looking at them at microscopes, they didn't have a lot of resolution, and they classified them as viruses. And it was only really until molecular work, like ribosomal RNA sequencing, that people were able to realize they were not viruses. So a third group is one of the two groups of gram-positive organisms, the phyla that include gram-positive organisms. Formally, this group is called the Actinobacteria. Informally, it's called the high GC gram-positives. And that the GC comes from people looking at the DNA of these organisms. And the DNA, remember there's A, C, T, and G in DNA. But if you have a C at a particular spot in the DNA, on the opposite strand, you'll have a G. If you have an A at a particular spot on the opposite strand, you'll have a T. So you can characterize an organism's DNA by its percentage of G plus C. So if it has 70% G plus C, it will have 30% A plus T. And these organisms in this particular phylum, almost all of them have very high levels of G plus C in their genomes. That's just where this informal name comes from. A lot of them form these very complicated, elaborate branching structures when they grow. And many of them were misclassified as fungi because this looks, as we will see later, a lot like the growth of many filamentous fungi. And again, it wasn't until molecular work in many of them that they were officially classified as bacteria in some cases. May not seem that important, but if you are infected with one of them and someone gives you an antifungal drug, it will not help. So you really want to know when you have something like this, whether or not it's a bacterium or a fungi. Many of the antibiotics that we actually use in medicine are derived from species in this group, like Streptomyces organisms. They live in the soil. They produce antibiotics to manipulate other organisms around them. And many of the antibiotics we actually use were isolated from this group of organisms. There are many pathogens in this group, including the causative agent of tuberculosis. That's an organism called Mycobacterium tuberculosis. Causative agent of leprosy, which is another member of the same genus, Mycobacterium leprae. And there are quite a few pathogens in this group, but quite a few soil and marine and other organisms. It's actually quite a diverse group. So there's a second phylum that includes gram-positive organisms. Its formal name is I call it the Firmicutes. Some people call it the Firmicutes, but I feel really uncomfortable saying that. And I don't even know which one is correct, but I just can't say Firmicutes ever again. And they are also gram-positive, again, with the same sort of general structure of the gram-positive organisms with a cell membrane and then a thick cell wall outside of that. Their DNA and their genome tends to have very low amounts of G plus C. So they are informally known as the low GC gram-positives. They are a distinct phylum from the Actinobacteria. But it appears that they either sister phyla or very closely related, and people have concluded that gram-positiveness evolved once in the lineage leading up to the common ancestor of these two phyla. It includes many pathogens are in this group, like the causative agents of anthrax, MRSA, the methicillin-resistant Staph aureus spreading around the planet, botulism, tetanus, etc. are in this group. Many interesting food-related microbes are in this group, like the lactic acid bacteria. And probably the most interesting biological feature of this group is shown in this picture here. So I mentioned binary fission before. So most of the time when bacteria or archaea reproduce, they make a copy of their genome, and then they partition the genome into two sides of the cell, and then they split basically right down the middle, and you get two cells, each with a copy of the genome from the parent cell, and the two cells are basically the same. Occasionally, in this one subgroup within the Firmicutes, I did it, you know? One subgroup within this group carry out this really interesting process where they basically make seeds to reproduce. They're called spores or endospores. And those endospores are basically produced in a deviation from binary fission. And the DNA is packaged up really tightly. This endospore is filled with all sorts of protection proteins and protection molecules. And then there's a coat put on the outside called a spore coat that makes them incredibly resistant to just about anything you throw at that. And then they wait around to germinate based upon some cue in the environment. The book discusses this a little bit. It reports what actually seems to be probably a real piece of information, that people think that they have been able to resurrect spores that are over 1 million years old. So they are very hardy, and that's unfortunate because this is why many of these organisms are used as biological weapons. Like anthrax, it's not normal growing anthrax that's used in biological weapons, it's the spores of anthrax that are incredibly stable. You can put them in a bomb, blow up the bomb, and the anthrax spores spread around and they have not been destroyed. That's why, tetanus bacteria and botulism, when you're making jam or something like that, you have to be really careful to boil the living, out of some of your samples in case there's Clostridium botulinum in there, because the spores of botulism can survive incredibly harsh treatments. Yeah?
So they die in boiling water?
They die in boiling water?
Maybe they die in boiling water.
To kill spores, generally you have to not only boil them, but boil them for a reasonable period of time. So most bacteria and archaea, except for the thermophiles we'll talk about later, but like the pathogens, E. coli, you can boil it for like 2 seconds and it's gone. But the spores of many of these organisms, you really have to boil it for like 10 minutes or sometimes even longer to really knock them out. They're incredibly hardy. You can throw radiation, like gamma radiation and UV radiation at some of these spores until the vessel that you're holding them in breaks, and they'll still survive. I mean, they're just completely absurd levels of resistance to various things that you can throw at them. A fifth group of bacteria are the Cyanobacteria. We'll hear a lot more about them because they are the source for chloroplasts, and that's the lineage that chloroplasts evolved from. All that have been characterized so far are photolitho-autotrophic, and they use a particular compound called chlorophyll A to capture the energy of light. Many of them have complex multicellular structures that they form, including some really interesting structures that are used for nitrogen fixation. So this is called a heterocyst that's used for fixing nitrogen. But many lineages within this group are multicellular. And we'll talk more about that when we talk about organelle evolution. So then the last group of bacteria, the last major lineage that we're going to talk about are the Proteobacteria. This is an incredibly diverse group. In fact, we can pretend for now that it's a single phylum, but it's probably actually composed of multiple distinct phyla within it. But let's just, we'll skip over that for now. The diversity within this group is extraordinary. There are photolitho autotrophs in here. There are chemoheterotrophs like E. coli. There are pathogens. There are mutualistic symbionts. There are extremophiles. There are just about everything you can find in here. They're all gram-negative. E. coli is in this group, so this group is incredibly well studied because of some model organisms as well as many pathogens like the causative agent of plague and cholera and typhoid and a lot of other diseases are in this group. This is just an example of one of the multicellular members of this group. And here's a nodulating organism in this group. We'll hear more about this. It's an incredible level of diversity just within this one lineage. The mitochondrion is derived from one of the subgroups in this lineage called the alpha Proteobacteria. And so we'll talk more about that when we talk about the origin and evolution of the mitochondrion. All right, that's your brief tour of the bacteria. Now on to the archaea. Are there any questions about the bacteria there? I know it's overwhelming, but I'm trying to give you sort of the big picture here. Yeah?
Is cyanobacteria gram-positive or gram-negative?
Cyanobacteria are gram-negative. And if you look at this tree, this phylogenetic tree here suggests that the gram-positive group the gram-positives are polyphyletic. So there's a group of gram-positives here, the low GC, and here, the high GC gram-positives. Don't trust the branching order here. So the phyla that we are talking about here all seem to be truly monophyletic groups. The Spirochetes, the Chlamydias, the Actinobacteria, the Firmicutes, the Cyanobacteria, the Proteobacteria. The branching order here is still the subject of much research and is not entirely clear. Most of the lines of evidence really do suggest that low GC gram-positives and high GC gram-positives are sister taxa, not like it's shown in this tree. And again, that gram-positiveness evolved once in the lineage leading up to the common ancestor of those two. But it is still, the branching order here is unclear, and you don't have to know anything about it because we don't know what it looks like. The archaea, there are two main lineages that we're going to... Did you have a question? Or?
Yeah. How does the high GC or low GC content of a cell, how does that affect the functionality, or does it?
So if you look at organisms when they have their genome, at any one point in time, it just has a set sort of GC content. But that content is actually under selection in a lot of organisms. So the genetic code is redundant. You can have 64 possible codons for only 20 amino acids. And in many cases, organisms prefer to use a specific codon even when they can choose from multiple ones. And the high GC gram-positives prefer to use the codons that have more GCs in them. So they're adapted to high GC content. And they stay that way, in essence. There's selection for their GC content. And the same is true for the low GC organisms. How they got there, to that point where they had high GC or low GC, is probably due to differences in how the DNA gets copied and just sort of spontaneous mistakes. So the low GC organisms seem to prefer, if all by accident, to put in A's and T's, and the high GCs accidentally put in C's and G's more, and that's how they ended up getting to the high GC content. It turns out there are a lot of subsequent effects of this. So, we're not going to talk about this a lot in the course, but high GC content organisms actually turn out to invent new genes more frequently than low GC content organisms because the stop codons in the genetic code are biased towards having A's and T's. So if you have a lot of G's and C's in your genome, randomly throughout your genome, you might actually have a coding region for a gene. It's more likely than randomly in an organism with high AT content. Just for those organisms, there are scattered throughout the genome lots of stop codons, so it's hard for them to invent a new gene because it'll get stopped when it's translated. So that may be why a lot of antibiotics come from the high GC gram-positives. There are a lot of papers on this. So there are biological differences. It's not clear what their origin is, but they're different. They have these different GC contents. Okay, so again, there are these two lineages of archaea, but like with the bacteria, there are actually quite a few more lineages of archaea than we're going to talk about here. This is from 10 years ago also. There are now probably 40 or 50 major lineages of archaea. There appear to be fewer major groups of archaea than there are bacteria known right now. It's unclear if that's sampling bias or really that there's lower diversity in the archaea. And if you look at this tree here, the Euryarchaeota is actually really better, would be called a superphylum. It actually includes many, many, many subgroups. We're just going to focus on the Euryarchaeota, but I just thought you should know that there's an enormous amount of diversity there. And the Creanarchaeota is another superphylum, and we're going to sort of completely ignore everything else. But there is a lot of diversity out there in the archaea, just like in the bacteria. So the Creanarchaeota, most of the taxa that have been characterized in the Creanarchaeota are both thermophilic, that is, they like high temperature, and acidophilic, that is, they like acid. So an example is this group Sulfolobus, where most of the species in this genus live at like 70 to 75 degrees Celsius and pH of about 2. That's basically warm acid bath. Ferroplasma is much more extreme. It lives at upwards of, sometimes 90 degrees Celsius and pH of 0. So that's basically a boiling acid bath. You can, that would kill almost everything else on the planet in minutes. And this is where this organism thrives, prefers to live in this environment. And we'll talk a little bit more about this when we talk about extremophiles. But this whole group of Creanarchaeota, many of them are thermophiles and acidophilic, although not quite all of them. The Euryarchaeota, we're going to talk about two physiological categories within the Euryarchaeota. One of them are the Methanogens. So many of the Euryarchaeota produce methane as a byproduct of their metabolism. So inside ruminants, for example, the cow digestive system, there are many methanogenic archaea, and that's why cows release a lot of methane from different parts of their body. And that is a major factor in global warming. Methane is a much stronger greenhouse gas than carbon dioxide, and a lot of the methane in the atmosphere comes from ruminants via their methanogenic archaea that are in their digestive tracts. There are quite a few members of this group that are also thermophilic, and many of them, the ones that have been studied, have been isolated in the bottom of the ocean at these cracks in the seafloor where high temperature water is coming out, called hydrothermal vents. The Methanogens are scattered throughout the Euryarchaeota. There's a physiological group within the Euryarchaeota called the Halophiles. These are organisms that love salt. And they actually correspond to a single monophyletic group within the Euryarchaeota. So they're just one of the branches of the Euryarchaeota, and every member of this group is what's called a halophile. And they prefer to grow in high salt conditions. We'll talk a little bit more about this again when we talk about extremophiles. Many of them are pink. So this is solar salterns over by the Dunbarton Bridge in the Bay Area. And people are evaporating the water to get the salt out. And as the water evaporates, the salt concentration goes up and up and up. And different organisms take over as the salt concentration changes. And when it gets to be really, really high, when you think nothing would want to grow in basically concentrated soy sauce, the halophilic archaea take over many of those environments. One of the more interesting things about the halophilic archaea is that they have, many of them have a protein that is unfortunately called bacteria rhodopsin. It was named before the archaea were discovered as a distinct group. And many of them have this protein. So you may be familiar with the rhodopsin in our eyes and other animal eyes. And the rhodopsin in our eyes serves as a light-mediated proton pump. So it sits in the membrane, takes the energy of light and pumps protons outside or inside of a cell. And you use that gradient to do things like see. The halophilic archaea do the same thing with this bacteria rhodopsin. It does not appear to be homologous to our rhodopsins. It appears to be a separate evolutionary invention of something that works in a similar way. Yeah?
Does that mean that the reason that we need light to see things is because light powers our eyes?
Well, I mean, we detect all sorts of forms of energy in the world around us, sound through our ears, and what our eyes do is they've adapted to detecting light, right? So that's how we see visually because we're seeing light. But people who are blind can sense the world around them still by hearing things and by touching things. So there are many different ways that we sense the environment. Visual light, that's how we defined it, is visual light is what we can see, right? There are other organisms that their eyes see ultraviolet irradiation. And there are many organisms that sense temperature, right? The heat sensors of snakes. So I don't know.
Well, the main thing I was getting at is that if we could find different ways to supply our eyes with ATP, would that mean we could see in the dark?
Probably not. So, but we can talk about that separately. What these halophilic archaea do is they use the energy of light to create these proton gradients, and then they use those gradients to make things like ATP. They are what are called photoheterotrophs. They do not fix carbon, so they are not photoautotrophs, but they use the energy of light to do some of their cellular functions. So interestingly, these organisms are an example of a species that is both phototrophic when it's using the bacteria rhodopsin and chemotrophic when they're living off of sugars in the environment around them. So that's the tour of bacteria and archaea. And I'm just going to give you a taste of eukaryotic diversity. Much more overwhelming. This is the list of the major lineages of eukaryotes in the book. I know you can read all of that there, right? So fortunately, this grouping is divided into 5 major subgroups. And what we're going to do, so Chromalveolates, you can see them highlighted here in the book. Chromalveolates, plants, excavates, and so on. And what we're going to do in the next lecture is go through these 5 major lineages and talk about their diversity.